Okay, so it's 1 minute past 9, so we should make a start. Welcome everybody to the JSPS Pathology Grand Rounds for February 2022. This morning it is my great pleasure to introduce Professor Fiona Watt to you. Fiona obtained her first degree from Cambridge University and her DPhil in cell biology from the University of Oxford. She was a postdoc at MIT, where she first began studying differentiation and tissue organisation in mammalian epidermis. She established her first research group at the Kennedy Institute for Rheumatology in London, and then spent 20 years at the Cancer Research UK London Research Institute. She helped to establish the CI UK Cambridge Research Institute and the Wellcome Trust Centre for Stem Cell Research. And in 2012, she moved to King's College London to found the Centre for Stem Cells and Regenerative Medicine. From 2018 to 2022, she was the Executive Chair of the Medical Research Council, and that position comes to an end shortly, and later this year she becomes the Director of EMBO. So it is my great pleasure to ask Fiona to speak to us today on exploring epidermal stem cell fate at single cell resolution. Thank you, Fiona.
Many thanks, Mark. It's a real pleasure to be here, and I will just share my screen. And there, that's okay? You can hear me? Good.
Yep, that's fine.
Fantastic. Well, this story I'm going to tell you today really spans some historical questions that my lab and others have been interested in for many years, and then brings it right up to date with the new technologies that we have available for looking at single cells in human tissues with resolution we never believed would be possible. So I feel actually ashamed at showing a pathology audience a section of human skin, but just to make the point, I'm going to be talking about the epidermis, but not the adnexal structures, the hair follicles and sweat glands and so on, even though they are really important. And I'm going to make the point in this slide that the way the epidermis is structured means that it is one of the classic tissues in which pathologists actually realize that there must be a stem cell compartment. And that's because cell division takes place in the basal layer of cells, here attached to an underlying basement membrane. And then cells that leave this layer don't divide any longer. They undergo a series of changes in morphology and gene expression, which culminates in the formation of these outermost acellular layers called the cornified layers. Although these cells are dead cells, they're incredibly important for our life. And as they're shed, they're replaced by proliferation of stem cells in the basal layer. And the epidermis is separated from the underlying connective tissue by that basement membrane that I mentioned. So showing the organization of human epidermis schematically, here are the stem cells. We've got cells which have left the stem cell compartment, and then the cells that differentiate as they go through the uppermost layers. And my introduction to the epidermis actually was as a result of being a postdoc with this man, the late Howard Green. Howard actually trained as in medicine, but he never practiced. And I believe that it was that medical training which made him realize very early on the practical applications of one of his discoveries. And that discovery was with his student, Jim Reinwald, that you can take biopsies of healthy human skin, disaggregate the epidermal cells, and plate them as single cells on a feeder layer. And when you do that, some of the cells will grow and form colonies. The colonies are multilayered with differentiated cells in the suprabasal layers. And over time, individual colonies in the dish merge, creating a multilayered sheet. And I still find it extraordinary that only 6 years after that initial discovery of the way of growing cells, Howard had led a team who had shown that you could, it's possible to graft burn wounds with autologous cultured epithelium. So this is one of the classic stem cell treatments. My interest, though, was in the stem cells, and my lab and others have used this ability of individual cells to form colonies as a surrogate assay in our search for stem cell markers. And even today, when we're screening different batches of culture medium to support the growth of human epidermis in culture, we plate the cells at clonal density, compare the different conditions, and simply count the colonies, as you can see on these dishes. And for a number of years, people were worried that this process was artificial, which of course it is. But in recent years, there's been some really fascinating analysis of clones generated in vivo, either with UV-induced mutations, for example, in P53, or what are called revertant clones in skin diseases. And this is just one example where you can see this particular disease manifesting as clones of cells which have got two disease-causing mutations on a background of healthy skin. So in the early work that we did, we used flow cytometry and surface markers as a candidate approach to see if we could enrich for clonogenic cells. And the first marker that we described, this is the work of Phil Jones, was high expression of extracellular matrix receptors known as beta-1 integrins. At the time, people really thought of integrins as being purely adhesive. But over the years, many groups have shown that these heterodimeric surface receptors regulate a huge number of different pathways downstream. And one pathway in particular, which has been characterized, is the classic ERK-MAP kinase signaling pathway, which I'll mention a bit later on. I'm not going to go through all of the candidate markers that we found, but I will mention another marker that was of interest, and this was work led by Sally Lowell, who now runs a lab in Edinburgh. Sally found that a ligand of Notch receptors, delta-like 1, was expressed more highly in clonogenic cells. The Notch signaling pathway, like the integrin signaling pathway, is well characterized. But I think what is particularly interesting about the studies that Sally led was that we realized that delta is doing very complex things within the basal layer of the epidermis. And those are summarized here. So we find clusters of stem cells in the basal layer of the epidermis that express high levels of DLL1, which is shown in yellow here. And the experiments that Sally initiated showed that expression of DLL1 actually protects stem cells from differentiating, but stimulates neighboring cells outside the stem cell cluster to differentiate. So we have a kind of cell-cell communication going on in the basal layer mediated by delta-like 1. And we also found that delta plays a role in clustering of cells and adhesion of cells, which is independent of Notch signaling. So really interesting and much more complex than simply a stem cell marker. To give you an update on some other work that we've been doing with characterizing cells in the basal layer of the epidermis, we were very interested in how it is that we could distinguish a cell which had committed to differentiate, so was no longer a stem cell, was destined to undergo this journey out of the basal layer, but was nevertheless not expressing any of the differentiation markers which are switched on in the spinous layers, which is shown here in purple. And we finally cracked this by going back to another classic Howard Green experiment, which is illustrated here. So back in 1977, Howard had shown that if you disaggregated cultured human epidermis and placed the single cells in suspension in a viscous medium, the cells, over a period of a few days, undergo a process of terminal differentiation, and by about 3 or 4 days, they formed cornified cells. So you can demonstrate this in different ways. We, of course, like to use our stem cell marker, beta-1 integrins, as a marker of the starting population. And if we track expression of beta-1 integrins or other basal layer markers, you can see that during the first 4 hours in suspension, this is qPCR, the levels remain high. But after 4 hours, from about 8 hours onwards, we see upregulation of spinous markers. This is one of the markers we use most frequently, involucrin. Here's another one, transglutaminase. And Jo Adams, who is a professor in Bristol now, showed when she was in my lab that something's happened to the cells at 4 hours in suspension, which meant that although they were still undifferentiated, they hadn't switched on involucrin, they were no longer responsive to ECM proteins or anti-integrins that would block differentiation. So moving to more recent times, Ajay Mishra, when he was a postdoc in my lab, did a wonderful collaboration with Tony Lee and Angus Lamond in Dundee, where we simply put our cells in suspension, as we've been doing for decades, collected them at different times, assayed colony forming efficiency. We knew that by 4 hours, colony forming efficiency had been lost. And then we compared the transcriptional profile of the cells with proteomics. And this was what Tony and Angus did using SILAC labeling. And what we found was that at this 4 hour commitment point in suspension, there were a lot of post-translational modifications in the protein data set, and these were dominated by protein dephosphorylation, something that, to be honest, we had never really considered at all. So based on integration of the transcriptomic and the proteomic data, we found that a total of 22 protein phosphatases were upregulated at 4 hours in suspension. And Ajay did this pretty heroic experiment of using siRNAs to knock down expression of each of the protein phosphatases individually, and then did a colony forming assay. So in this spider diagram, each blue circle represents 10% of the plated cells forming clones. The control, the scrambled control, is red, and you can see that just over 20% of the plated cells formed colonies. And then if we look around all of the different knockdowns, we can see that if we knock down some of them, we actually increase colony formation. For example, this one, PTPN13 or DASP6. And there are a few, such as this one here, where knocking down the expression reduces colony forming efficiency. And just to show you something else that Ajay found, we found that in contrast to, for example, the basal layer markers, which go down after 4 hours, or the differentiation markers, which go up after 8 hours, our protein phosphatases tended to peak at 4 hours and go down subsequently. We've done a lot of studies to characterize some of the key protein phosphatases. And one of the things that we found, which was very interesting, was that if we use a whole mount antibody labeling process to visualize the basal layer of the epidermis, we can see clusters of our putative stem cells, which have high levels of beta-1 integrins, and you can see them as these little peaks or hillocks of cells in the basal layer of the epidermis. Some of the markers, like DASP6, are beautifully co-expressed in those stem cell patches. Other ones show a complementary distribution, like PTPC7, and some of them, like DASP10, show a more uniform expression. So we not only have these genes, which when they're upregulated are promoting differentiation and driving cells out of the stem cell compartment, but we see a complex and highly patterned distribution. We know a lot about the signaling pathways which regulate the onset of differentiation. And in work that we did to characterize the effect of the phosphatases, we found that they impact on ERK-MAP kinase signaling, so that's downstream of the integrins. And just to illustrate that, this shows the top 15 dephosphorylated peptides that Tony found in the data set, and actually 3 of them correspond to ERK-MAP kinases. So there's no doubt that these are important in differentiation. And then keratinocytes, like most cell types, express a lot of AP1 transcription factors, many of which control expression of differentiation genes like involucrin. And we were able to show that the phosphatases impact on AP1 signaling. So far, so good. We were quite pleased with that. But we did start to notice that we should not be considering these phosphatases individually. And this was because we found that when we knocked down expression of one phosphatase, it typically affected expression of another. And furthermore, that those interactions were very dynamic. It depended, the interaction and the effect depended on where the cells were in suspension. So just to talk you through this, we're setting the level of the RNA of this set of 6 protein phosphatases as baseline at 0 hours in suspension. Then if it increases or decreases from that point, increases shown in red, decrease in blue. And then the arrowheads show a positive interaction between expression of individual phosphatases, whereas the T-bars show a negative interaction. And what we were able to do was collaborate with Sarah Jane Dunn, who was then at Microsoft in Cambridge. And she took our data, our experimental data, the RNA levels and the protein levels, and performed a Boolean network analysis. And this led her to model the data as a situation in which the stem cell state is stable, the differentiated state is stable, but this transient upregulation of the protein phosphatases acts as an inherently unstable commitment state. And we could use various pharmacological treatments to artificially stabilize the stem cell state or the committed state to provide further evidence for this hypothesis. The studies that we did in suspension give us very good temporal information, but most recently, we really wanted to look at differentiation of individual cells. And to do that, we were able to work with a fluorescent sensor, a FRET sensor, which gives you the level of ERK signaling activity in individual cells. This is the work of my former postdoc, Toru Hiratsuka, who had trained in Mishi Matsuda's lab in Kyoto. And it was the Matsuda lab, which has the expertise in FRET sensor technology. So we introduced these constructs, the construct into cultured human keratinocytes. It reads in the nucleus, which is really convenient because we could combine that with a fluorescent reporter of our differentiation marker, involucrin. So we put the FRET sensor and an M-cherry reporter of involucrin activity into the cells and simply monitored what happened over a period of hours. And I'm showing you here 4 individual traces of 4 individual keratinocytes. And in each case, you can see over time the fluorescence increases as the cells differentiate. But what we noticed was this very interesting pulsatile ERK activity, which preceded the upregulation of involucrin and a subsequent decrease in basal ERK levels. So we did a lot more analysis to work out what was going on here. And in particular, we were able to show that if we artificially increase the levels of 2 of our phosphatases, those impact ERK basal activities and ERK pulsatile activities. So this is a doxycycline-induced vector, which increases DUSP6 or DUSP10. Increasing DUSP6 has no effect on mean ERK levels, but it does decrease the pulses, whereas DUSP10 has no effect on the pulses, but decreases mean levels. So to cut a long story short, our current model of exit from the stem cell compartment is that it occurs through this unstable pulsatile ERK activity, and that our protein phosphatases are regulating this, with DUSP6 regulating the ERK pulses and DUSP10 reducing ERK levels so that exit from the stem cell compartment becomes over time irreversible. I'm not going to read all of the conclusions from this slide because I've shown you the data, but I would just mention in passing that we were able to actually image these ERK pulses in the epidermis of anesthetized mice. So we know that in that live animal model, this pulsatile activity is taking place. So as I'm sure you know, over time, we've become really obsessed with our hypotheses, our experimental models. We tend to meet other people who've got the same interests. And if I dare say it, sometimes it seems a little bit cozy. All of that, I believe, changed in 2016 when Aviv Regev and Sarah Teichmann launched a global initiative, which is called the Human Cell Atlas. And this has the ambitious goal of mapping all the cell types in all of the tissues in the human body. And back then, the aspiration was that we would get new insights into human physiology and disease mechanisms. That was one goal. And the other goal, which I think is equally important, is driving new ways in which we can integrate different data sets, collaborate, and share data. So I'm going to tell you now how the Human Cell Atlas has impacted on our work on the epidermis and skin more generally. For those of you on the call who are interested in the sorts of techniques that are being used, I actually took this very nice summary diagram from a review which was based on single cell profiling in the kidney. But essentially, if you have a tissue like skin or kidney where you can obtain a biopsy, you can disaggregate the cells, then you can use different techniques to, in particular, 10x technology, to sequence the RNA transcripts in individual cells. You can then use that to provide maps of clusters of cells that seem to be similar. And there are techniques in which you can also map the spatial arrangement of the cells in the tissue, which of course is really exciting too. We were very fortunate to join a team led by Sarah and Mazlifa Hanifa in Manchester, funded by Wellcome, to do quite a large-scale mapping. And our work at King's has been led by Magnus Lynch. So in work which was published almost a year ago, we provided the first human skin cell atlas data from our collaboration, although I should emphasize that they're beautiful data sets from a number of other labs that we look at actively too. So we had healthy skin from 5 adult donors. These were mastectomy specimens, adult mammoplasty. Maz had some data from fetal skin and also from 4 patients with atopic dermatitis or eczema, 3 patients with psoriasis. These were mixed age and sex, but what was really good was that for each individual we had non-lesional and lesional skin. So you can imagine this has been a really happy hunting ground for all of us and really very interesting to analyze. So of course we have 2 predictions from our work over the years in cultured human epidermis. We predict that there should be multiple populations of basal layer keratinocytes based on our work on the stem cell compartment and stem cell markers. And we believe that there should be, it should be possible to identify cells that were committed to differentiate because they were expressing this network of protein phosphatases. So what we've done most recently, and this is one of the things that I'd like to emphasize, is that maybe classically you think once you've published a paper that's done and you move on. But when you have access to the single cell RNA sequencing data, you can publish the data set and then download it, blend it with other data sets and really keep the analysis going because it becomes much more of a living data set, if you like. And what we were able to do, and by we, I mean Victor Negri, who was a PhD student in my lab and now works for AstraZeneca, was to take the big data set that we had published, go into it and look specifically at those 5 healthy adult donors and look just at the epidermis. And a great advantage that we have working in the epidermis is that we know we've got lots of markers for the basal layer, the spinous, and the granular and the cornified layers. And so Victor was able to confidently assign basal markers based on expression of basal keratins, spinous markers based on suprabasal keratins. And we also had a small number of cells which we knew had come from the granular layer because they expressed the marker filaggrin. So what did we find? These are UMAP plots. So this is very, can I say, flexible data. It's looking for clusters. The more cells you put in, the chances are the more clusters you will see, because you'll be able to identify smaller clusters which were not present in the bigger data set. And it seems blindingly obvious, but if you don't disaggregate all the cells to do the analysis, they're not going to appear in the plots. So I think we're used to looking at sections and being pretty confident about what's going on, but these UMAP plots are very useful, but they're very much a sort of snapshot of the single cell data. Anyway, with that health warning, what we found very clearly was within the basal layer, we could see these three distinct populations, which Victor's just simply named basal 1, 2, and 3. So what is in there? Well, it was really interesting for us to be able to look. We were able to have a look at our existing stem cell markers from our group and from many other groups. And we could see that all of the markers we looked at mapped to the basal layer, but interestingly, their relative distribution in those clusters differed. So beta-1 integrin, our favorite stem cell marker of all time, was most highly expressed in basal 3, whereas CD46, which by flow cytometry is co-expressed on the surface with beta-1 integrin, is actually more abundant in basal 2. And our favorite stem cell marker in the notch pathway, DLL1, which is expressed, frankly, in almost homeopathic quantities, was nevertheless, we had a real signal there in basal 2. Other markers, caveolins, which we know in culture are co-expressed with the cells with highest levels of DLL1, were most abundant in the basal 1 population. And this marker, which is validated by Wang et al., you can see is most expressed in basal 2. Here are individual plots just showing you how the markers map to the different layers. And so what we can say is that we're confident that there are three different populations of cells that we can see in the plots, that our stem cell markers map to them, but their relative abundance differs. So what we're currently doing in the lab is to ask whether the three populations are equivalent or functionally distinct, and whether or not they're arranged in a hierarchy. Can any cell get from basal 1 into the spinous layer, or does it have to go through the other basal populations? So that's the stem cell markers. What about the protein phosphatases? Well, here, using expression of the markers that we had from our cell culture experiments, we were able to identify two cells populations, which Victor's named transition 1 and 2. And we could indeed see expression of our markers in there. You can see that DASP6 expression is more abundant in transition 1. That's also here on the UMAP plot. And interestingly, we could also see some expression down here, which is basal 2, which is where we see, basal 1, where we see high levels of beta-1 integrins. And DASP10 is stronger in transition 2 and also is more widely expressed. And I thought this was particularly interesting because if we go back to the qPCR we did of cells in suspension, we always noticed that the DASP6 peak was much tighter than the DASP10 peak. And then our work with the FRET sensors would argue that they're impacting on differentiation in different ways. So really interesting and thought-provoking for further experiments. So to summarize what I've said here, we can say with confidence that the single-cell RNA sequencing has confirmed at least some of our experimental predictions. But really, what we want to do is move on from that. And we can ask, can we now use the data to generate new testable hypotheses? And I'm just going to tell you about two pieces of work that we did. to answer questions that we've been interested in for a long time. So one of the questions is, given that DLL1 is expressed in such low levels, and it's distinct from other Notch ligands in that it keeps cells in the stem cell compartment rather than stimulating differentiation, is there something that we've been missing about the way this Notch ligand works? And then, of course, the other thing we can do is have a look at how the stem cell populations are altered in psoriasis, again, going back into the master data set and pulling the keratinocytes out. So to answer the first question, first, this is again Victor's work. It's been known for a long time that there's a group of fringe proteins which are N-acetyl glucosyl transferases. And their role is to transfer N-acetyl glucosamine onto O-fucose residues in the EGF repeats of the Notch receptor. So the Notch extracellular domain has many EGF repeats. And these enzymes change the way that Notch is glycosylated. There are 3 fringe proteins in mammals, lunatic fringe, manic fringe, and radical fringe. That you can actually date these to the time when Sonic the Hedgehog was all the rage in the studies in game consoles. But what we did then was to look in the single cell data set and see, yes, indeed, we could pick out lunatic, radical, and manic fringe. That lunatic was most highly expressed of the 3, and furthermore, co-expressed with in basal 2, where DLL1 expression is highest. We knew from the biochemical evidence that lunatic fringe could selectively increase the affinity of Notch receptors for DLL1 and inhibit the responsiveness of Notch to the ligand jagged. We used qPCR from samples of human epidermis, which were not from the data set, to validate the findings. We couldn't detect manic fringe at all, and lunatic fringe was expressed more highly than radical fringe. If we look in culture, again, we could see some upregulation of manic and radical, but lunatic fringed remains the most highly expressed, particularly in the culture conditions where we grow cells at clonal density. So the next question is, if we modulate expression of lunatic fringe, can we modulate the stem cell compartment? And I'm just going to show you experiments in which we use the lentiviral vector to overexpress lunatic fringe in cultured human keratinocytes. We carried out a number of different assays, for example, looking at whether or not differentiation markers were downregulated. And as this series of qPCR shows, yes, indeed, most of the markers that we looked at, like involucrin, envaplakin, and so on, were downregulated on overexpression of lunatic fringe. But I think what was perhaps the most striking was that if we did our colony forming assays, you can see that we got a big increase, not only in the total number of colonies, but in their size. These colonies are much larger than we would see normally and would tend to support the concept that lunatic fringe expression is supporting stem cell maintenance. So the other question that we asked was, what about the keratinocytes in psoriasis and eczema? And here we've taken Victor's 13 keratinocyte cultures, looked in the data sets at how their distribution is changed. So the first thing to say is that we could find, and we could account for all of the disease-associated keratinocytes amongst our 13 clusters. But you can see that the relative proportions are quite different between psoriasis and eczema. And actually, you can't see it in this slide, but it is pretty clear that the psoriasis and eczema non-lesional epidermis is quite different in the distribution of the different clusters than we've seen in healthy skin. And one of the things we know from the histology that the spinous layer is expanded in psoriasis, but it's really intriguing that in psoriasis, we see a really selective expansion of this population of cells, which Victor has named spinous 4, which we don't see, for example, in eczema. So again, very thought-provoking and lots that we can do. And this just is a bar chart, which where we take all of the cells that we've got in the data sets and just try to illustrate how the proportions of those different clusters change. Now, again, we have to be careful because the numbers of cells that if we don't isolate all of the cells, that's going to skew the results that we get. And again, we need bigger numbers of cells to be able to be confident about the smaller populations. But nevertheless, I think this difference in the spinous layer compared to lesional and non-lesional, lesional and non-lesional epidermis and between psoriasis and eczema is very clear. And this relative reduction in the size of the basal 2 compartment is very striking if we look here compared to the other populations. So lots and lots of food for thought and more experiments that we can design. So to summarize what we found so far, it's been good because we've been able to confirm the existence of multiple stem cell populations, supporting the conclusions that we'd made from years of carrying out experiments with cells in culture. It has, as I've illustrated, provided a really strong platform for designing more mechanistic experiments, and it is already starting to provide new insights into disease mechanisms. If I have one concern or plea with all of this, it is that we need to make sure that as biologists or pathologists, we get sufficient training in bioinformatics that we're able to do at least the base level analysis of the data sets ourselves, because I fear that if we don't keep up with this sort of technology, we're going to end up being de-skilled. Once you have the tools and the data sets to look at, it's really very straightforward to just at the click of a button answer a question about a particular gene of interest, and of course integrate across different tissues. So I'm almost finished with my presentation, but I did want to end with a look into the future. And this is really telling you where my lab is going next. And I would say that our interests probably reflect the way the field is going as a whole. So I told you that we had integrated transcriptomic and proteomic data sets for keratinocytes with time in suspension. But I'll illustrate how we've taken this one further step and integrated it with lipidomic data sets. And I do believe that looking at lipids and carbohydrates is incredibly important functionally. So this is important for us. Fibroblasts have been quite a strong interest of our group for a number of years, so I'll mention that. And then finally, I love the idea that you could simply run a probe over the surface of somebody's skin and be able to see in real time how the proportions of the different cell types are changing. So I'm going to end by just summarizing these 3 current interests. So the work that we've done on lipidomics is the work of Matteo Vietri-Rudun, who's a postdoc in the lab. And Matteo needs to be congratulated for recently publishing an amazing compendium in which we tried to review all that we know about lipids in the epidermis. It is quite a read and has been quite fun to put together. But we were fortunate that we were able to have 2 collaborations for this project. One was with Ricky Eckert, a group leader at King's, who allowed us to use a platform she has for knocking down lipid modifying enzymes in keratinocytes and looking at the effects on differentiation. And the other was a collaboration with this company in Germany, which provided the lipidomics work for us. They're not like your average company because as well as providing the data, we got an extensive critique from the CEO on how we should have designed the experiments and what the flaws were in our analysis. So that has been fun too. So the critical thing to remember with the lipids is that it's not like, RNA makes protein. You can have lipid modifications which are catalyzed by a number of different genes, and one enzyme can catalyze multiple changes. So it is really complicated, and compared to single cell RNA sequencing, I think the lipidomics field is some way behind. But nevertheless, this was what we were able to do. We were able to, for example, here look at the ceramides and how those increased with time in suspension, and look at the effect of inhibiting protein kinase C, which blocks differentiation, stabilizes the stem cell compartment. So you can see that blocks ceramide induction. And equally, if we look at hexosyl ceramides, we see a similar effect. Then we can do unsupervised clustering, just as you can do for RNA and protein, to look at the changes in all of the lipids with time in suspension. And here we've done a principal component analysis, so where we're mapping our different populations of cells. And what was interesting was that we could cluster the most differentiated cells together, the starting population over here. And then here, where we're looking at commitment and the effects of the inhibitors and early differentiation, we're seeing some similarities that we need to work out. But again, the data set is published, and I think is very interesting as we start to think about how lipids, intracellular and on the plasma membrane, impact on classic signaling pathways. Moving on to the fibroblasts, this is a schematic from a few years ago where we were highlighting that if we look at the skin of a mouse, we can define a number of different subsets of fibroblasts based on location and function. So cells which control the hair growth cycle, cells which form a smooth muscle that controls the level to which the hair shaft is sticking up from the surface of the skin, and then layers of papillary, reticular, and hypodermis. And this was a sterling work by a number of postdocs, but in particular Ryan Driscoll, and combining lots of different assays that you can do readily in a mouse, in particular lineage tracing. We published the concept that during mouse skin development, there's a common fibroblast progenitor. And then as you go from E12.5 of mouse development to neonatal life to P2, you can see a number of, we would argue, differentiation decisions, which at different times in development lead to the formation of the multiple cell types that we see in the dermis. Although this was a huge amount of work, it turned out that it's by far from over. And in particular, the markers that we use to flow sort different populations of cells are really dynamically expressed, and they're definitely not fibroblast specific. And so we moved on from this to bulk RNA sequencing data, trying to compare mouse with dermatome sectioned human skin. And that is pretty much where we landed in 2018, confirming that we could see in human skin that there were different populations of fibroblasts and getting some hints that they have different functions. So of course, now we can go back into the big data set that we and others have generated. In the data set that we published last year, we could see 3 distinct fibroblast subpopulations, but we actually believe by combining it with more data, the number of fibroblast subsets is probably greater. It's more like 5. But again, it's very interesting. We can distinguish them based on markers that would confer function. For example, here, discriminating markers involving metalloproteinases and collagen genes. Lots and lots that we can do. And then, of course, we can look in psoriasis and eczema and look at how the relative proportions of fibroblasts and indeed vascular endothelial cells and other cell types change. And so here, if we look at those 3 fibroblast populations in healthy skin, then if we look in non-lesional, lesional eczema, and non-lesional and lesional psoriasis. Again, we can see that those three populations are present, but their relative abundance changes in disease, that there are differences between eczema and psoriasis, and also that non-lesional skin is distinct from healthy skin. So a wonderful resource to look at that further. And so now I'm just going to end with the final thing that we've been doing, and it's to speculate that it would be really great if we could integrate all of these different methods that we've been using, so that we could, by running just a probe over the surface of the skin, we would know what cell types are present. The technology that we've been using is this optical coherence tomography. It's called a Vivo site. It's made by this company. This shows you how it works. It's literally just ultrasound on the skin. And we can look at the skin in different body sites non-invasively. We can also use plugins which allow us specifically to visualize the vasculature of the superficial skin. And here we can look specifically at the thickness of the epidermis. And we know that in psoriasis, the epidermis is thicker than in healthy skin. And here you can see an example of the different thicknesses that we can pick up. So just as this rotates, obviously that's the surface of the skin, that's the epidermis. You can see the basal layer because of the pigments of the melanocytes. So we know where we are there. And actually we can read the dermis by seeing the blood vessels and using the changes in the extracellular matrix to distinguish the papillary from the reticular dermis. So again, lots of data that we can use. And just to give you a taster of where we've got to, this is work from Clarice Garnier, postdoc in the lab, where she simply lined up lots of volunteers in the lab and looked at the differences in vascular density between different body sites. So, I won't take you through it, but it's pretty obvious here that the level of vasculature in the forearm is a lot lower than in the lip. And we would really like to know what the reasons are for those changes, because we can combine this analysis of different body sites with the single cell RNA sequencing data, and that's work in progress. So that's me at the end of my presentation. Just to summarize, the single cell RNA sequencing data sets confirm the existence of multiple populations of cells in the epidermis and the dermis. They provide an important reality check for our experimental models, and they have indeed given us new insights into normal skin physiology and inflammatory disease. I haven't mentioned basal cell carcinoma or squamous cell carcinoma, but the data sets are there. And the fun now really is trying to pull all of these different types of data in space and time together. The last slide is actually the most important. This was, I think it was July last year. We thought that the country would be out of lockdown and we'd be able to meet inside. But this was the time when you were allowed to have up to 30 people outside. So it was supposed to be a game of rounders, but the pleasure of a reasonable day and being together socially was great. So I would like to thank them for just being wonderful and sticking with it during what's been a very tough time for scientists all over the world. So with that, I'll stop sharing and I'd be happy to take questions.
That's fantastic, Fiona. Thank you very much for a really interesting lecture. Absolutely brilliant. Can I ask the audience to put questions into the question and answer box, please, and I will read them out for Fiona. So we'll start out with a question from Simon Harrington, who asks, are the multiple epidermal basal cell populations present in squamous and basal cell carcinomas?
Could you ask me that next week? So we and others have. So Paul Kavari's lab has got great SCC data. We're just bringing in the BCC data, but we just haven't squished it all together. So I'm going to bet, and it's a bet, that they will be there, but what will be different is the proportions. And in case anyone asks about fibroblasts, cancer-associated fibroblasts, we're looking at that, and the initial results are suggesting not a new population, but an expansion in BCC. So sorry, Simon, but it's a great question.
Okay, thank you. So the next question is from Guy Betts, who says, thank you, that's really interesting. Do you think the basal 1, 2 and 3 compartments are geographically distinct or all coexist within a single maturation unit?
Guy, that is a great question. We have good evidence at the protein level that the cells with the highest levels of integrin, CD46, delta are clustered. But what I forgot to mention actually is that the protein turnover time of the different cell surface markers is different. So at the moment, the cells that are co-expressing those markers at the protein level are clustered, but we could get more resolution within the clusters using fish probes, for example, and that's something that we need to do. And in terms of a single maturation unit, if you're thinking about that beautiful early work from Chris Potton and Ian McKenzie, I think that is specific to those parts of, for example, a mouse ear where the columns are well arranged. We see the units much bigger in the epidermis. And yes.
Okay, thank you. So Walter Bodmer asks, what actually triggers the start of differentiation?
Thank you for that question, Walter. We know from classic studies from people like Iversen in Oslo, Leblanc in Canada, that it's not caused by neighboring cells, mitotic cells squeezing cells out of the basal layer. What we think is that as the cells become committed to differentiation, their anchorage to the basement membrane is downregulated and their affinity for suprabasal cells is increased. So we could, I speculate sometimes that if you've been out of cycle for a long time and you're getting bigger, your proportion of the cell surface occupied by the basement membrane is reduced, and that could trigger the start of differentiation. I think what I've learned actually is that the epidermis is pretty robust and has lots of checks and balances to stop everything differentiating. So it will be a combination of different factors.
Okay, thank you. So John Tadros asks, have you looked at whether microbial transcriptome, the microbiome, affects the single cell transcriptome either proportionally or in terms of cell cluster DEGs? Is it something that could be co-assessed?
That is a great question. Our lab has done really nothing on the microbiome, but there is some beautiful work. Primarily, I would say I would attribute to Yasmin Melcade and Julia Segre at the NIH, where they were really pioneers of showing that the microbiome is different in different body sites. And we now know that the proportion of skin cells is different in body sites. So it is just a matter of data integration that you could correlate changes in the microbiome with changes in the proportion of cells, for example, in the epidermis. And I did not mention T cells or other cells of the immune system, but of course, all of that data is available. And so you could correlate changes in the microbiome with changes in the immune system.
Okay, great, thank you. So David Katz asks, in terms of the focus on psoriasis, can you yet look for genetic polymorphisms that are associated with the expanded yellow population, which I think was the spinous 4 population?
Yeah, yes. We haven't done that. We could do that because, of course, we've got great collaborators around the UK who specialize in polymorphisms associated with psoriasis. Chris and in Manchester and others. But I was thinking, actually, as I was preparing the talk, we need to go back and see what is that yellow population. I don't know if we've got specific markers for that. And then, of course, we can, assuming we have probes for it, it would be a simple piece of work to look for that change. And again, would love to do it.
Thank you. So Tim Fenton asks, do you know what is driving the spikes in ERK activation in the transitional cells? Is EGFR signaling acting upstream to oppose the dephosphorylation by the phosphatases you discussed?
Yes, we published some information about EGF in Toru's paper about the spikes, which came out in 2020. The weird thing about EGF and keratinocytes is that, of course, EGF is added to the medium to promote proliferation, but high levels of EGF will drive differentiation. And so what we could see was that a high level of EGF can be upstream of the spikes and actually promote the dephosphorylation events. But if you go for different levels, then you'll see, I didn't mention it, but if cells show pulsatile activity in a high base level, they will be able to go back into the stem cell compartment, for example, after proliferating. And I also didn't talk about it, but it's fascinating that although we found our protein phosphatases by using proteomics, they're transcriptionally, the levels of the transcripts are really tightly correlated. So there's so much going on there. It's mind-boggling.
Okay, thank you. So next, Andrew Dean asks, can you comment on what molecular differences distinguish hair-bearing from non-hairy skin, such as chin versus lip? And do they give insight into mechanisms of disease in conditions that respect those boundaries?
That's a great question. We don't have data sets for, lip or palmar skin yet. But the class, it's known classically that the Hox code is respected in adult skin, and there are particular keratins that are only expressed in those regions. And actually, a few years ago, Inish Sequeira and I published a study where we looked at a keratin called keratin 76, which has a highly patterned distribution, and it's linked to, in the mouse, to loss of that keratin is linked to a specific increased risk of cancer in the oral cavity in the forestomach. And we see changes in the lip, including something which I think you would call smoker's lip, where for reasons we still don't understand, you get a line of melanocytes appearing at that border. So lots that could be done, but for that, I would probably try integrating the mouse boundary data work with the human, because it's pretty well conserved.
Okay, thank you. So in the last minute, perhaps I can ask a question myself, and that is, you described some very elegant experiments on the network of phosphatases and emphasized the roles of DUSP6 and DUSP10. I just wondered what you know about what are the targets of those phosphatases that they actually do change and dephosphorylate.
I showed you that en masse they can affect ERK, but that's only part of the story. P38 MAP kinase, which is important for differentiation, absolutely targeted by some of them, and some of them, or at least one of them, is associated with notch signaling. So you've got multiple pathways present. And I think going back to this robustness of protecting the stem cell compartment, you kind of, it's like pushing different parts of a spider's web. You can touch over here and there'll be an impact over there. Really interesting.
Thank you very much indeed. Now, we've just hit 10 a.m. and we do have a tradition of finishing on time when we can. So with that, I will say once again, thank you very much indeed, Fiona. That was a really fascinating talk. Thank you very much.
My pleasure. Thank you. Goodbye.
Thank you.
Bye.